Hello and greeting to Dr. Suganti and my fellow friends. We are now presenting to you microorganism and its biochemical application in natural resource. So, we are choosing Methanococcus as our bacteria for our assignment. But before we start, let me introduce you to our teams. Hello everyone, so today I would like to talk about our topic, which is microorganism and its biochemical application in natural resource, and we choose Methanococcus as our assignment. So it's my desire to introduce you a captivating topic from microscopic realm, Methanococcus. Let's dive right in. Methanococcus, as we know, are non-pathogenic archaea bacteria thriving in strictly anaerobic, hydrogen-tropic condition. These incredible microorganisms are commonly found in marine environment. Now, let's talk about their temperature preference. Methanococcus is acquired the temperature connoisseur, showcasing a range of preference. Some prefer moderate temperature, earning them the title mesophilic. One of the most intriguing aspects of Methanococcus is energy metabolism. As strict anaerobe, they produce methane as the major product. This process, known as hydrogen-tropy, involves utilizing hydrogen gas to generate methane, contributing to the global methane cycle. Methanococcus, like other archaea, possesses an information processing system that more similar to eukaryotes than bacteria.
Next, Methanococcus has gram staining that is gram-positive or gram-negative because it is an archaeon. In addition, this Methanococcus does not have a definite cell wall structure that can distinguish it from bacteria, and one of them is because Methanococcus is often found in extreme environments such as in high temperatures. As we know, methanogens are those that produce methane by consuming carbon dioxide and organic molecules. Methanococcus can be found in types of anaerobic environments. These habitats are characterized by low availability of electron acceptors due to the activity of consortia of fermentative bacteria. These fermentative bacteria decompose complex organic matter into simple fermentation products.
Now we are going to look at pictures of Methanococcus. There are three pictures of Methanococcus that we include here. Figure 1.1 shows Methanococcus bacteria. Figure 1.2 shows Methanococcus meripeludis cell, and Figure 1.3 shows Methanococcus janashi. Next, methane producing genera pictures. Methanogens are artibacteria that produce methane as metabolic byproduct. Like Figure 2.1 shows Methanobacterium. Figure 2.2 shows Methanosarcina. Figure 2.3 shows Methanospirillum, and Figure 2.4 shows Methanococcus.
Biotechnology application of methanogens. We have four types of biotechnology application of methanogens. Firstly, biogas methane production. Second, mitigation of methane production from natural source. Third, carbon capture and utilization. And fourth, model system for hydrogenotropic methanogenesis. Biogas methane production is a renewable energy source. In anaerobic digester, microbes convert organic waste into methane, a combustible gas used for electricity generation, heating, and transportation. Reduce greenhouse effect compared to fossil fuels. The second point of biotechnology application of methanogen is mitigation of methane production from natural source. Mitigation of methane production from natural source. It helps clean wastewater by anaerobically degrading organic matter and produce methane. Environmentally friendly, reduce the need for chemical treatment methods. It can be a renewable source energy and enhancing sustainability.
Next, I will explain about carbon capture and utilization. Methanogens have been engineered to produce valuable chemicals such as coenzymes, isoprenoids, and carboxylic acid. Next, it helps capture atmospheric CO2 and convert into a valuable products. Methanococcus maripaludis has been designed to capture CO2 from the air and turn it into valuable products, offering a simple yet impactful solution to reduce atmospheric carbon dioxide and great useful substance. For the next topic, I will talk about model system for hydrogenotrophy methanogenesis. The chosen model system for hydrogenotrophy methanogenesis offers a valuable platform for researching microbial metabolism, genetics, and enzyme function due to its rapid growth, ease of manipulation, and well-annotated genome. This model has significantly contributed to advancement in biofuels production, environmental remediation, and our understanding of the global carbon cycle.
Now, we are going for biochemical pathway of applications. As you can see in the slide, figure 3.1 is Methanococcus maripaludis cell. Figure 3.2 is methane productions, which focus on metagenesis. Next, figure 3.3, methane cycle. This picture shows various sources for the productions of methane. Next, we are going to go through the biochemical pathway of applications of Methanococcus productions of methane. There are 5 steps involved, which are hydrolysis, acidogenesis, acetogenesis, methanogenesis, and lastly biogas productions. Now, we're going to look into one by one of the process. Hydrolysis is a complex organic matter such as carbohydrates, lipids, and protein that broke down into simple compounds through hydrolysis by microorganisms. Hydrolytic bacteria produce enzymes to facilitate the conversions into monomers like sugar, fatty acids, and amino acids. Second, acidogenesis. Monomers are future metabolized into organic acid by acidogenic bacteria. Involves the fermentations of sugar and other organic compounds.
The third part of biochemical pathway application is acetogenesis process. It is start with acetogenic bacteria that convert into acetic acid, which is produced in the acidogenesis phase. And it is important intermediate step before methanogenesis process occur to produce a biogas production. Moreover, we are going to talk about methanogenesis process, Methanogenesis starts with Methanococcus, that being a methanogenic archaea, and it is play an important role in the final step of methanogenesis. In this process, it converts acetate and other one-carbon compounds into carbon dioxide and methanol into methane, which is CH4. Methanogenesis also is carried out by specific enzymes in Methanococcus, such as a methyl coenzyme M reductase, or we call it as MCR. And lastly is biogas production. As we know, methane gas is produced by Methanococcus that, along with trace amounts of carbon dioxide and other gases, biogas. In biogas production, it can be captured and utilized as a renewable energy source for various applications, including electricity generation and heating process. Next, as you can see on this slide, this is the key and important steps in methanogenesis process, which are: firstly is formation of acetate, secondly is acetate formation, and thirdly is methanogenesis. In methanogenesis process, you have to remember that there is three processes that occur, which are formation of methanol, reduction of coenzyme M, and lastly is release of methane gas.
Then we go to the next slide, which is a case study on the application. In this section, we get 4 journals where the journals are online journals. Case study number 1 is about function and regulation of the format dehydrogenase genes of the methanogenic archaea Methanococcus maripoladis. The focus of the case study is on the genetic and biochemical aspect of Methanococcus maripoladis, especially its ability to produce methane from hydrogen plus carbon dioxide and format, as well as the study of the format dehydrogenase gene. Case study number 2 is about genetic and metabolic of Methanococcus. In this case study, say Methanococcus is a well-studied genus of mesophilic and hydrogenotropic methanogens known for their robust genetic system, typically found in tidal marshes. These microorganisms compete with sulfate and iron-reducing bacteria for their primary energy sources, hydrogen gas and format. Thriving in mineral medium, Methanococcus exhibits rapid growth and requires marine level salts for optimal development. Over the past 2 decades, genetic tools such as shuttle vectors, CRISPR systems, and marker proteins have been refined, enabling researchers for functionally characterize the genome. This includes studying the regulation of the transcriptome and proteome, mapping transcription start and termination site, and identifying essential genes. The development of metabolic and gene regulatory network models based on these foundations knowledge opens up possibilities for biotechnological application of these hydrogenotropic methanogens.
Next, the rest of published paper that we have found related to Methanococcus. The first article is a review, renewable and sustainable energy reviews. It's explained about anaerobic digestion of municipal solid waste with methane production by Methanococcus maripoladis. Next article is International Journal of Environmental Research and Public Health. In this article, it's explained about bioremediation of heavy metal contaminated soil by Methanococcus voltae. The third article is International Journal of Hydrogen Energy. In this article, it's explained about production of biohydrogen by Methanococcus maripoladis from agricultural biomass. Next article is Journal of Renewable and Sustainable Energy. In this article, they explain about biofertilizer production using Methanococcus species for sustainable agriculture. And the last article is Bioresource Technology. This article explains about methane production from biomass by co-culturing Methanococcus maripelidis and Clostridium termacellum.
Reference. These are the references that we use to complete this assignment. There has 5 references that you can see.
That's all from us, Group 8. Thank you for spending your time throughout the presentation.